Okay, so as you noticed, we are moving on now to Unit 4A, talking about DNA. The test on Friday, from what I heard, had some issues, so I don't really have a lot of good data on it quite yet. Um, but we're done with plants. We'll stop talking about plants, and you'll probably tired talking about plants. So we're done with all that. We are now moving on to genetics. So genetics is going to be our final unit. We might have one more unit at the end about like genetic engineering because that's a new thing on the STAR they like to talk about. But really, genetics is going to be, that's part of it. It's going to be our last unit, our last big unit. So it's February, which means that we have about 3-ish months to the STAR. And I think if we're counting everything correctly, we have like 10 or so weeks before the STAR. My math is not great on that, but the point is, we don't have a whole lot of time before the STAR. So this is our last unit. We're going to try and get through it as quickly and efficiently as we can. That way we'll have enough time to review before the real thing. I think at this point, assuming everything stays on schedule, we should have at least 2 weeks dedicated to review, maybe even 3 weeks worth of review before the STAR. But okay, so today's objective is to describe the monomers necessary for cells to build DNA. So for those of you writing in your journals, I'll give you a second. I'm sorry, not your journals, your planners. Okay. Alrighty, then let's get into it. Oh, I forgot to replace this. So as far as the CK through 12 reading and the do now, I apologize, those are from last week, but you'll figure that out pretty quickly. All right, so you guys are going to be group D virtual. I don't know if you guys heard, but there's been a slight change to the schedule, for the most part impacting people here in person, not really impacting you guys. Okay, so I'm going to give you a few minutes to go ahead and start working on that do now, and I'll be checking. I think we're waiting on a lot of people to get in. Most of what I'm seeing is from my last class. Thank you, Avery. Thank you, Brighton. Thank you, Dominique. Thank you, Faye. Thank you, Hugo. Thank you, Josiah. Thank you, Cameron. Thank you, Clint. Thank you, Melanie. Thank you, Miguel. Magia. Hey, look, there's me. Thank you, Terrell. Thank you, Zaria. Good job, you guys. Make sure you're getting on to class if you're working on your do now. Oh, Avery, I guess I should check. Let me check. Ah, I don't know if you noticed, it was there for a second and then it blipped off. That's crazy. Like here on the preview, like I saw that there was stuff written, but then as soon as I went into it, it was blank. But don't worry, I know that you're working hard. So thank you for doing your best. About 30 seconds left. I hope you all had wonderful weekends. I hope you got lots of relaxation and that you are ready to face the week. Okay, let's talk about the do now. Oops, don't look at that. Oop. Yoop. Anybody feeling particularly confident about number one? What are the three components of a nucleotide?
I'll give it a shot.
Go for it.
Okay, the three nucleotides. There's adenine. Uh, there's guanine. I don't know how you say that. It's kind of hard, Mr. Martinez.
That's true, but I think if you were looking over to the left, you might see three components of a nucleotide underneath structure of a.
Oh yeah, right. It has nitrogen, a 5-carbon sugar, and a photophase group.
This is too big. Let's go. There we go.
Whew. Mr. Martinez, those are some hard words to say on the right.
Oh, it's only going to get worse. So this is the basics, right? We got the three components. We got our nitrogenous base, we got our 5-carbon sugar, and our phosphate group. Those are the three parts of a nucleotide. And remember that nucleotides are going to be the building blocks of DNA. Alrighty, so we talked about the building blocks of DNA a long time ago when we talked about the different biomolecules, right? We talked about lipids, carbohydrates, nucleic acids, and proteins. So nucleic acids were those genetic ones. The building blocks, right, the monomers of a nucleotide, the DNA is going to be a nucleotide. Okay, very good. Thank you, Brendan. All right, who thinks they can list the nucleotide bases present in DNA?
You said the DNA, right?
Mm-hmm.
Okay, so let's see here. DNA.
Well, I already heard from you once, Brandon. We answered number one together. So how about somebody else tackle number two?
Oh yeah. Let's let you go. Try it.
Okay.
Or Josiah already put in the chat box what you just said. DNA is a RTCG.
You got it. Very good. Thank you, Josiah. So the nucleotide bases present in DNA are going to be these over here. So adenine, guanine, cytosine, thymine. Thankfully, the star is not really big on making you spell stuff out, right? So don't worry about the spelling as far as what these are. But A stands for adenine, G stands for guanine, C stands for cytosine, and T stands for thymine. These are the four bases that make up DNA. So everything about who you are, your skin, your hair, your eyes, your brain, your muscles, your bones, all that stuff is coded inside your DNA and is one of these four letters, right? Those traits are combinations of those four different letters. Okay, so that's DNA. Now, RNA, a little bit different. Can anybody tell me the difference between the nucleotide bases for RNA and DNA? If you don't want to come off mute, you can just write your message in the chat box like Josiah did and Avery did earlier.
Did you want me to help you read the chat box, Mr. Martinez?
That's fine. I can see both.
Okay.
Mr. Martinez, can you hear me?
I can hear you.
Okay. Is it the AGC and Uracil? Uracil?
Very good. So almost identical. The only difference is that instead of a T, you have a U. Oh no, Hugo, your camera's on. Please turn your camera off. Please turn your camera off, Hugo.
Wait, sir.
Just kidding.
I got so scared. You
I
literally
know.
scared me. I like covered it with my hand and everything.
Okay, so instead of thymine, there is uracil. Now, a big difference between RNA and DNA. RNA is meant to be temporary. So DNA is going to be all of your traits, right? Half your mom, half your dad combined together to form you. So that's going to be your DNA, all your DNA. That's your traits, that's you, everything. Now, your RNA is going to be like little short messages. So let's say, for example, that I pull some hair out and I got to make some more hair. When my body needs to make that hair, it's going to find the DNA sequence that codes for my hair. So it finds that code, makes a copy, and then it does its business. So that's going to be an RNA copy of your DNA. So RNA is not meant to stick around for that long. They're more meant for short messages. We'll talk more specifically about DNA and RNA later, but for the most part, when it comes to structures, these are going to be the biggest differences. So we talked about the ATCs and Gs. Now we're going to be looking at the sugars present in DNA and RNA. So does anybody know what the name of the sugar is in DNA?
Deoxyribose.
Very good. Thank you, Eileen. I think that was Eileen. There you go. Deoxyribose. That's what the D stands for in DNA. It's deoxyribonucleic acid. So again, we're talking about genetics, we're talking about genes. That's going to be nucleic acid. So the D stands for deoxyribose, which is the sugar that makes up DNA. So that's how you know what DNA or the nucleic acid that we're talking about. So if DNA is deoxyribose, what does that make RNA?
Ribose.
Very good. So if DNA is deoxyribose, RNA is going to be ribose. And that's it. There you go. Simple as that. That's called ribonucleic acid. Okay, so we got our differences, right? We figure out what's structurally different between them. If you're interested about chemical structures in the sugars, you can see here we have an H versus this hydroxide found in ribose. So it's missing the oxygen in the deoxyribose, which is why it's deoxy, right? That's missing the oxygen. That's more biochem stuff, which we'll talk about in AP Bio and or biochemistry. Okay, so what are the biggest differences then between DNA and RNA? I'd say the first big difference, RNA has uracil instead of thymine. Two, the sugar for RNA is ribose. The sugar for DNA is deoxyribose. RNA is temporary messages, which we'll talk about later. Okay, so those are going to be our biggest differences between DNA and RNA. And this has been a refresher of nucleic acids. Like I said, we talked a bit about this when we were talking about biomolecules, but it's been a long time. So we're going to review it and now deep dive into it. Okay. All right. DNA structure. Can I get a volunteer, please, to read this first paragraph for me?
I can read it for you.
Thank you, Zaria.
DNA structure. DNA has a double helix structure that consists of two polynucleotide chains. The two helices bind to each other, forming a double helix via weak hydrogen bonds between complementary nitrogenous bases. What is that? Adenine?
You can just say the first letter. You can just say A binds with T.
Oh, A binds with T, and G binds with C.
Very good. Thank you, Zaria. Okay, so let's talk about the structure of DNA. So think of DNA as like a ladder, right? So here's a ladder with the rings. I know that ladders usually go up and this one's going sideways, but just bear with me. Oof, there we go. Ladder, right? So when we're talking about a double helix, what we're talking about is this ladder that's been twisted. So it's turning like this. So imagine that the ladder was made of like something pliable, like clay. You took the two ends and you kind of turned them like this. And that's how you get this DNA shape. So you take those two sides and you twist them around each other and you get that shape. And you still have the things in the middle. So we've talked about DNA before, right? We've talked about these nucleotides. We've talked about the sugar phosphate backbone. This is what that all comes out to, is this. So here, you'll notice that we have, let's call it sugar phosphate backbone. Let's call this one pink, because I like pink. White. Nice manly color, right? So the sugar phosphate backbone is essentially what's running along this edge. So sugar phosphate, sugar phosphate, sugar phosphate. So those are the things that are running along the sides. So like that.
Okay,
That's what's.
Mr.
Go ahead.
Martinez, how did you draw that?
Draw it.
What you just drew?
I use this pen tool right here. Okay, and now I'm going to draw it again because the highlight kind of takes all the room out of it. So the sugar phosphate backbone, if you were paying attention to the shape of a nucleotide or the components of a nucleotide, that's going to be 2 out of the 3, right? So we have our sugar, phosphate, and then we have our nitrogenous base. Let's call it blue. Specifically A, T, C, and G, right? So let's use our highlighter and get some blue action. These then are those nucleotides, the stuff that's going in the middle. So whenever we're talking about DNA code, what we're looking at are those structures in the middle, the stuff that actually makes up the DNA code. So let's say, for example, this is a trait for, I don't know, brown eyes, right? So it would be like A, T, A, T, T, A, G, C, whatever, right? Those are the things that link together. Specifically, we have, let's make it white, and a color you can see. A is going to bind with T. Ooh, I said bandwidth. How about bind with? B binds with C. The easy way to remember this, if you guys recall, apples under trees, cars in garages. Ring any bells? These are the Chargaff's pairing rules, which we're going to talk about today. Okay, so the double helix is wrapped around histone proteins and supercoiled to make it more compact in the nucleus of eukaryotic cells and the cytoplasm of prokaryotic cells. Now, what that means is that DNA takes up a lot of space, right? Look at this stuff, right? It's kind of all over the place. So imagine like a yo-yo, right? A yo-yo has a nice long string. So this string takes up X amount of space, right? Let's say that yo-yo then is curled up around a little thing. Look at how much space there is between these two things, right? Here a lot of space, here's not as much space. So the DNA does the exact same thing. The DNA essentially coils around these circles and becomes super compact. So whenever we're talking about like, chromosomes being XX or XY, it's because they are all coiled really, really tightly and they form these structures. I can probably do this better. How about like this and like this? So whenever we're talking about those X's and Y's, we're seeing those shapes in nature, but those shapes are created by that DNA wrapping around these histone proteins. It's a lot easier to draw this on a whiteboard. I hope you can appreciate that. Okay, so the histone proteins make it possible for it to supercoil and make it nice and tiny. So think of the histone proteins as like the yo-yo, right? And the string as being the DNA. So the histone protein is wrapping and wrapping around, so it takes up a lot less room. If the DNA was not supercoiled, our bodies would be like as big as the Earth because there's just so much information there, right? Like each one of these chromosomes has the entire data for you. And so they're super, super, super coiled. Like it's crazy to imagine how much supercoiling is going on. I think I read somewhere a long time ago that if you stretched out your DNA like one molecule at a time, it would stretch all the way from the Earth to the sun. Like it's a crazy amount of information. But okay, so that's a double helix. Okay. Any questions before I move on? I see no questions. I will move on. Now, brief history lesson. The star likes to bring up this junk right here, which is X-ray diffraction crystallization. The important thing to remember is that we didn't know a whole lot about DNA until pretty recently. So right around the 40s or 50s, what happened was this lady, her name is Rosalind Franklin, was shooting X-rays at different structures to see if she could find out what they look like at an atomic level. So whenever you go to the doctor to get an X-ray done, what happens is they put let's say that you broke your arm, right? You hold your arm over that plate, they shoot the X-ray at it, and what happens are these supercharged particles bounce off of certain areas, right? And it gives them a better picture of what's going on inside your body since, we can't see past our skin. So the same thing happens on a very molecular level. They're shooting all these supercharged particles at molecules, and they're seeing where the molecules are. Because again, too tiny, right? We couldn't see this with a microscope. Even now, you need what's called an electron microscope to see DNA. So these are things that are kind of beyond sight. But what they were doing is that they were using these principles to see if they could visualize this atomic world. So Rosalind Franklin was using all this radiation stuff, and she was basically figuring out the structure of DNA. She didn't really know it at the time, but that's what she discovered. Here you can see the helix kind of wrapping around, going that direction. So what happened was these two guys, Watson and Crick, pretty much saw her data and stole it, for better for worse. Now, there's a lot of different accounts of how this happened, and I think for the most part, everything is... I think it's pretty straightforward, but I think that's when you kind of cross the line between history and opinion. So I'm going to teach you just what I heard. So what I heard is that Watson and Crick, who are these two guys, pretty much stole Rosalind Franklin's data, and they were able to kind of form an idea of DNA. Now, again, they couldn't have done it without her, and she really wasn't looking for that. So they kind of use each other for mutual beneficiation. Oh, I guess no. Watson and Crick used her data. She didn't get anything out of this. She was messing with DNA at a time that we really didn't understand DNA. This was like in the 40s. So she actually died, many years before Watson and Crick got like the Nobel Prize for figuring out the structure of DNA. So her contributions for a long time were kind of forgotten and kind of pushed aside because she was a woman. But in the last decade or so, I think we've seen a big push to recognize Rosalind Franklin as being pivotal in finding out the structure of DNA. So again, the reason I bring this up is because the star does like to show you this picture and wants to know that you know that this is showing you the structure of DNA. They're not necessarily quizzing you on who Rosalind Franklin was. If anything, they'll probably just tell you. But they are going to show you this image. I can guarantee it and expect you to understand that this is looking at the structure of DNA. If we had more time, I would have you write this, but we're running out of time, so I want to skip this. Boop. Okay. Investigating the building blocks of DNA. Can I get a volunteer to read this for me, please?
I can.
Thank you, Josiah.
Erwin Chargaff,
Chargoff.
an Australian biochemist, had read the famous 1944 paper by Oswald Avery and his colleagues at Rockefeller University that demonstrated the heredity units or genes are composed of DNA. This paper had a profound impact on Chargaff, inspiring him to launch a research program that revolved around the chemistry of nucleic acids. Of Avery's work, Chargaff wrote the following. This discovery almost abruptly appeared to foreshadow a chemistry of heredity and moreover made probable the nucleic acid character of the gene. Avery gave us the first text of a new language, or rather he showed us where to look for it. I resolved to search for this text. As his first step in this research, Chargaff set out to see whether there were many differences in DNA among different species. After developing a new paper, chromatography method for separating and identifying small amounts of organic material, Chargaff reached two major conclusions.
So we're going to be looking at that today. So whenever you're going through these big blocks of text like this, especially when it's using really, really high academic vocabulary, which by the way, Josiah, fantastic work. Good job. It's important to make sure that you are kind of keeping track of what's going on. Like when I was your age and even up to now, when I'm looking at this big block of text, I can get lost in what it's talking about. So I think it's very helpful to make sure that you're highlighting, especially on the star, the major points, like what's being said, right? So what's happening here is that there's a scientist, and he was really interested in genes, right, and the composition of genes, specifically like nucleotides. And that's proved here with the nucleic acid character of the gene. That's essentially talking about nucleotides. Nucleotides. So this scientist studying genes is trying to figure out basically how they work, right? And it wants to look at the differences in DNA among different species. Okay, so the hardest part I think about genetics is that we have a lot of math. The math here is pretty easy. The math later is going to be a little bit more difficult, but for now we're just dealing with pretty much simple math. So looking at this table, I think you can start to notice a pattern. So take a second, look at the data. Tell me if you see any kind of pattern in your data. Anybody notice any patterns? What if I told you that for the human, we had 30 A's, 30 T's, 20 G's, and 20 C's?
Sir, can you hear me?
I can hear you.
You said we're looking for a pattern, right?
Mm-hmm.
Okay, is the pattern that A and T's are usually the same because apples and trees, and then G and C's are the same because of cars and garages?
You got it, man. Very good. So, thank you, Hugo. Remember that A binds with T, C binds with G. So if we're looking at these mathematically, like these are ratios, right? If 30% of this DNA in a human are going to be A's, then it would make sense then that 30% are going to be T's, right? Because they're going to be the same. Because A is binding with T, so you're going to see the same numbers of A's as T's. So A is 30, T is 30, C and G are 20, 20. So what that means is you add all these up, 30 plus 30 plus 20 plus 20 is going to equal 100%. So let me scooch over here. Actually, I'll scooch over here. So 30 plus 30, that's going to equal 60. And
Hey, Mr. Martinez, sorry about that. My Wi-Fi went out.
that's okay. 20 plus 20 is going to equal 40. Then you do some more math. That's going to equal 100.
Okay, but Mr. Martinez, but why is there a chart to let us know which one goes in first?
If we had some more time, I'm running out of time, we're not going to do this. If we had more time, I would chart this and we would see the pattern visually with some data, but we're running short on time, so I'm going to skip that part for now.
Okay.
What I want you to notice, though, is the pattern. So there are the same numbers of A's as there are of T's, the same number of G's as there are of C's. So this is a math problem that you're going to encounter on the star. So here we're given a data table with some blanks, and we're going to try and figure out what the blanks are. So let's start with wheat. Let's go ahead and round 27.3 to 27. So we got 27 A's. How many T's then are we going to have? If 20% are A's, how many percent are we going to see of T's?
27.
Very good.
Yeah.
Right? They're the same. How about C and G? Actually, it's G's first.
G, you're gonna see 22%.
Let's round it up to 23.
Okay.
Which means that for C, you're also going to have 23. Now, in math, you're gonna learn something called proofs, which is essentially to know how something is true, right? So we were given 27 for A. So 27 plus 27 is going to equal 54. Now, if you take 54 and divide it by 2, if you take a 54 and subtract it from 100, you're going to get what's left over. So 100 minus 54 is going to equal, I'm going to scooch this over just a little bit, 46, right? So 54 is going to be A plus T. 46 then is going to be G plus C, right? Because that's what's left over. So 54% is going to be the percent of A's and T's. That means that 46% is what's left over for G's and C's. Now, if you wanted to figure out specifically which is which, you would take 46 and divide it by 2 to get your individual ones, right? So 46 divided by 2 would be 23 and 23, respectively. Okay, have I lost anybody on the math yet? Good. I'm going to take that to mean that everybody's still with me. Just to make sure that we are clear on that, I'm going to go ahead and include that here. So 42 is going to equal 23. Okay. All right. So the math is not difficult. It's just got to think about it a little bit. For the most part, the star is not going to be asking you to do anything super crazy. It's going to be addition, subtraction, multiplication, and division, like we just saw here, right? We're adding some numbers together. You're dividing a number by 2. That's it. When it gets to predicting offspring, that's going to be a little bit more risky because that's more mathy stuff. But this, just dealing with percents, not too bad. Okay, now let's look at yeast. So yeast, we're given, I think, 33 T's. And I'll get rid of this one too. I guess I might have a faster. It's just type it. Lazy. Okay, so yeast, we have 33 T's. Which means we're going to have 33 A's. So 33 plus 33 is going to equal 66. So 66% of this DNA's or of the organism's DNA is going to be A's and T's, right? So then you take 100, you subtract 66, and what does that leave you with?
$34.
Very good. So now 34% G's and C's. So you take that 34%, divide it by 2, and what do you get?
Divided by 2?
Mm-hmm.
17.
Very good. And as you can see here on the chart, even though we rounded, right, 17.7. So it's not 100%, but you get the point, right? The pattern is relatively stable given something that's organic, right? Like this is not a computer doing this. This is your body. Now, we talked about it with the cell cycle, that sometimes your bodies can make mistakes, right? That's where cancer comes from. Because when we're talking about DNA, we're talking about your body replicating that DNA from the previous cell, right? That replication. We'll talk more about that later. But your body can make mistakes. Sometimes mutations, sometimes cell cycle errors, whatever, right? That's why sometimes the numbers aren't 100% there, because again, we're looking at real numbers. If we're dealing with the concept remains the same, right? All these numbers should add up to 100. Okay? So we got wheat, we got yeast. Can someone solve E. coli?
And what does that mean?
So we got these blanks here for E. coli. What are those blanks going to be?
E. coli. It's going to be 24.
Very good. So we got 24A. So we got 24A. How many T's do we have?
We only got still 24?
Very good. So if we have 24% A, that means that 24% T is what's going to be there. So you take 24
plus 24 will gives you 48.
Very good. Then you take 100 minus 48, and that's going to give you the percent of C's and G's, which is what?
100 minus, let's see, 100 minus 48 plus 148 minus 4, 52.
Okay, then you take 52 divided by 2, or you look over here with G being 26, you divide 52 by 2, and I think you'll find out that it's 26.
Yep.
And there you go. That's it. Okay, so the math, like I said, is not difficult. There's a lot of adding, dividing, subtracting, right? Pretty easy stuff. Now, when it comes to the star, they're usually going to show you one number. So this one's been kind of nice because they showed you two numbers. And the real thing, they're probably only going to show you one. They'll give you one number. They'll say if there's, 30% A's, how many C's are there going to be? And then you got to figure out the math yourself. But we just did it. You got the formula right there if you want to memorize it, which is not necessary. It's just basic math. But there you go. Okay, so are there any questions about this? Man, you guys are just on it. Okay. Boop. So let's do some check for understandings. Draw a DNA double helix with the following code: ATT, AGC, CGT. Boop. Remember that DNA helix is pretty much a ladder, right? So I'm going to go with a ladder instead of it being all twisty. So we've got actually I'm going to do it typed while I'm doing it written. ATT, oh, space bar even more. ATT, AGC, CGT. Those like to be spaced out more. Okay, so that's our DNA code. Remember that A binds with T, T binds with A, G binds with C, C binds with G, there we go. So here are the DNA nucleotides, right? Now let's finish out with the rest of the DNA double helix. Let's say that we got this here, we got this here, this here, this here, and then whoop. You know what, we can probably go even bigger with this. Yeah, there we go. Yeah, so again, this is going to be our sugar phosphate backbone. The nucleotides, actually, I guess I should make that better. How about we go like this? Woo, woo. And then we have our hydrogen bonds in between our nucleotides. And there we go. There we have a pretty simple DNA double helix. So remember, it's just like the rings of the ladder that we are twisting to make it look like this. And the stuff that's inside are going to be these nucleotides. Okay, next. How do nucleotides bind together? What holds the DNA, strands, should be strands together. So, nucleotides are held together by weak hydrogen bonds. I should have mentioned this earlier, but I guess I'll mention it now. The DNA ladder is held together by the sugar phosphate backbone. So the sugar phosphate backbone is very strong. The hydrogen bonds, though, are very weak. Why do you think it would make sense that the hydrogen bonds between the nucleotides are weak? So again, the stuff holding the ladder together, pretty strong. The stuff holding the nucleotides together, pretty weak. Why do you think it's important that the nucleotides are not held together too tightly? Any ideas? What if I did this? Oops. Ugh, that was ugly.
So like to prevent it from making more DNA strands?
Not prevent. I would say to facilitate. So we talked about the S phase in the cell cycle, right? The DNA has to make a copy of itself. If the DNA is held together super tightly, it's probably pretty difficult to make a new strand, right? Because remember, to break that strand apart takes a lot of effort. So if there's hydrogen bonds there that are weak, you can remove them pretty easily, and then you can make your new DNA strands. So the reason that they have weak bonds so that they can break apart during DNA replication. Which is also the S phase of the cell cycle. So again, the DNA, the backbone, very strong. The rings in between, those where the nucleotides are bound, those hydrogen bonds, those break easily so that when it comes time for DNA replication, the DNA can be broken apart pretty easily. They can make a new copy, then it can right back. So everything ties back to something else, right? So we're talking about genetics, but this is going back to our cell cycle concepts from a long time ago. It all comes together. Okay, and last, what type of biomolecule is DNA RNA? What elements do they contain? So the biomolecule, of course, is going to be nucleic acids. The elements, cho cho chon chop, right? So remember, CHO are going to be carbs and lipids. CHON are going to be proteins, and then CHONP are going to be nucleic acids because the P is going to be part of that phosphate group. Okay? Ugh. So much information, right? Okay. So if you're feeling overwhelmed or confused, please ask questions. If you are wondering if we're going to be skipping over stuff and talking about different things, we're going to revisit this stuff tomorrow. So we're still talking about DNA structure, and we're going to start looking at DNA replication. So we're going to go back and look at the cell cycle one more time. But we're still talking about DNA. So if you feel lost, don't worry. We're still talking DNA for a long time. Quick reminder, thank you, Nick. I don't know if you're in here. If you have not turned in your plant project yet, your plant project was due on Friday. So if you haven't got that turned in yet, please get that turned in. That'll help your grade immensely, assuming you did well on it. Okay, we've got a little more than 10 minutes, which I think is probably pretty good. I guess I did have time for the chart. If you want to go back to the chart and graph it, I would highly recommend it because it's very helpful in looking at the trends in data. So if you finish your eggs ticket, go back, finish this. I guarantee that it'll make you draw some connections. In the meantime, though, the rest of you, go ahead, start working on your exit ticket. Once you've completed your exit ticket, you can go back and fill out that chart. Or if you have not finished your plant project, you can definitely get that turned in to me. But yeah, so you're taking the rest of our time today to work on exit ticket, to work on your plant project, and to fill out that chart. So 3 things. First, tackle that exit ticket. Then you can work on the chart or your plant project. Okay, any questions? We got about 10 minutes. So I'll go ahead and stop the recording.
Mister, was there a right way to do the plant project?